virtual screening could identify SAM competitive inhibitors. It also indicated that inhibitors could outperform RG108. And the mechanism of these inhibitors are very different than 5-ASA, which has been approved by the FDA, as I mentioned. However, again, 5-ASA is incredibly toxic. So our hope at that time was that we could identify inhibitors that were less toxic but had equal or enhanced efficacy compared to 5-ASA. And that's the path we're on. I'll come back to that in a little bit. The other enzyme that we became interested in is G9A, and we began to ask the same questions. Could we use virtual screening to identify compounds that can inhibit G9A? And there's a bit of a story to this, and I'll let you know in a little bit. But G9A is a histone 3 lysine 9 methyltransferase. It can add 1, 2, or 3 methyl groups to lysine 9 of histone 3. And we became interested in asking whether or not we could identify compounds that could specifically inhibit monomethylation, dimethylation, or trimethylation, or some combination of these. And the reasons are a little complicated, but one reason we became very interested in trying to find monomethylation inhibitors of G9A is that as G9A activation increases due to overexpression or whatever, G9A, histone, lysine 9 of histone 3 becomes monomethylated, and that actually activates serine glycine pathway. This is a pathway that produces macromolecules, and cancers obviously grow rapidly, divide, need lots of macromolecules to be produced. And the thought was, if we could identify an inhibitor that specifically inhibits monomethylation, and we caught the cancer early enough that we could actually inhibit this reaction and decrease or turn off serine glycine pathway. And I'll show you some data down the road that this is something that we are able to do. As the cancer progresses, di and trimethylation events occur. This typically turns off tumor suppressor genes. So the other question is, can we target di trimethylation activities and restore expression of tumor suppressor genes? And I'll show you some data for DNMT1 that actually we can turn these genes back on. So again, we started answering our questions. That is, could we identify first compounds that specifically inhibited the enzyme that we're interested in? Turns out G9A is not the only histone 3 lysine 9 methyltransferase. Another enzyme is called GLP, does basically the same thing, and SUB39H2. But each of these enzymes sort of have different functionalities. And so the question is, even though they all add methyl groups to lysine 9 of histone 3, could we identify compounds, at least virtually, that could distinguish between these enzymes? And this is just a quick look. But in general, about 50 to 60% of the compounds that we identified seem to be specific in terms of their interactions with the enzyme of choice. So in this case, 62 compounds were specific for G9A when it's complex to SA, 61 compounds GLP complex to SA, and 53 compounds for SUB39H2 when complex to SAM. There certainly is some overlap, but again, this is very encouraging to us that we could identify, we thought we had a basis to move forward to identify compounds that specifically interacted with the enzyme that we were interested in. The other question that we asked is that these enzymes tend to complex with different substrates. And so the question is, could we identify compounds that were specific to the enzyme and whatever substrate it was complexing with? In this case, we're looking at GLP complex with SA, GLP complex with SA and H3K9 monomethylated, and G9A or GLP complex with SA with dimethylated H3K9. And again, much to our surprise, the vast majority of compounds that we were identifying, at least virtually, were specific for the enzyme and the types of things that it was complexing with. And so that gave us some encouragement to continue to move forward. We wanted to know why that might be the case. So again, we started doing some more computer-based types of experiments, I guess. And so we asked ourselves, why would a compound interact with the binding site of G9A and GLP? I don't know what your backgrounds are, so I want to make sure I'm saying this clearly. It turns out that the SAM binding site of these molecules is very flexible. And that flexibility gives the opportunity to produce lots of different types of structures. And so we started looking at the interaction of compounds within these enzymes. So for example, again, G9A and GLP carry out the same reactions. In this case, we're looking at a compound interacting with G9A when it's complex with SA, and a compound interacting with GLP complex with SA. And what you should see is that even though this is the same catalytic domain, you can see very differences in terms of structure. For example, here's kind of a bulbous region that doesn't exist over here in GLP, and it's because of these differences we think that we can identify compounds that are very specific not only to the enzyme, but to the enzyme complex with other substrates. So again, all this was encouraging for us, and we started doing some bench work, and in this case we were asking early on a question: could our compounds inhibit monodi and trimethylation reactions or one of these reactions? In this case, our compound 0148 inhibits monodi and trimethylation reactions of G9A. 179 only inhibits di and trimethylation reactions, same with 260. And then 318 inhibits only the trimethylation activity. And so, again, this was early on, but very encouraging to us that we could identify compounds that could actually inhibit the specific activities of this enzyme. We think that there's application for that's going to be important. And I'm not going to show you data, the recent data, but we do have identified compounds that can actually do this, and we're looking at these now in terms of their ability to alter the function of these cancer cells. I won't bore you with this. I don't know how much biochemistry you guys loved in the early days, but we obviously have to demonstrate that these compounds actually are competitive inhibitors. These are classic competitive inhibitor curves of some of our compounds. This is another compound, BIX1294, which is also a G9A inhibitor, but it is not a competitive inhibitor. Its craft is very, very different. So in terms of cancer, one thing that's becoming realized is that histomethyltransferases have an association with a lot of cancers, and this is just a partial list of them here. As I mentioned, we're focusing on G9A in cancer, and it turns out G9A is overexpressed in a large number of cancers, including liver, gastric cancers, gynecologic cancers, head and neck cancers, leukemia, prostate cancer, lung cancer, as well as breast cancer, and they're mechanistically linked to metastasis. It turns out when G9A is overexpressed either in lung cancer or breast cancer, this actually disrupts the extracellular matrix, and these cells begin to wander. There are no therapeutics targeting G9A at this time. I think that we're in competition with one other group, but again, they go after G9A inhibitors in a different way than we do. As I mentioned, we're targeting triple-negative breast cancer. This is a cancer that represents about 15% of all breast cancers, 10 to 20% of invasive breast cancers. TNBC cancers, or triple-negative breast cancers, are characterized by a lack of estrogen receptor, progesterone receptor, and HER2 expression. When these cancers metastasize, survival time is typically not longer than a year. There are not good treatments for it. There's high medical demand to develop treatments for triple-negative breast cancer. Typically, the treatment surgery and some very strenuous chemotherapy and radiation therapy. There have been some therapies looked at in the clinic, but pretty much non-efficacious. And so G9A really is a new target for triple-negative breast cancer, as well as other breast cancers. As I mentioned, many cancer cell types rely on activation of serine glycine pathway. There's a biomarker for this pathway, PHGDH, or phosphoglycerate dehydrogenase. In these ER-negative breast cancers, about 70% of these cancers are characterized by gene amplification of PHGDH. The other cancers, PHGDH is overexpressed. So one question we had is, could we develop a G9A inhibitor that could inhibit G9A and either silence or turn off the serine glycine pathway? And one way to assess that is looking at the expression levels of PHGDH in these cell types. Again, this is just an overview of what I said, but the idea is we can develop a G9A inhibitor, and it will knock down, turn off this PHGDH portion of the pathway. It also will probably turn off other portions of the pathway, but this is really what we look at. And in this case, this is just one piece of data demonstrating that one of the compounds that we've been working with actually does inhibit G9A and reduces levels of PHGDH significantly. And we've compared that to another G9A inhibitor, again called BIX. This is the compound that BIX will never see a clinical trial, but it also reduces PHGDH levels, but our compound does it much more significantly. You take this out a few days, we'll see this go up and down, and it's still always better than BIX. And so again, this is really encouraging to us that not only we can identify these G9A inhibitors, but we can use them to hopefully silence a pathway that's turned on and can reduce the functionality of that pathway. This is just some more data indicating that TDH levels are reduced when treated with the G9A inhibitor, and we can also demonstrate that H3K9 monomethylation is reduced in response to this G9A inhibitor. Our current efforts right now are to continue to optimize our compounds, and we're using X-ray or crystallography to do that. We've been able to develop crystals of G9A complex to a variety of things, and our main efforts right now are to complex our compounds to G9A and get a real sense of how this compound's interacting with the enzyme and potentially how we can improve the compounds. This is just our operational plan. Right now we're here. We think we can get an IND submitted by late 2019. We've just signed a contract for about $7 million in funding, about half of which is going to go to this G9A program. It turns out that we've been doing some work with Jackie Stevens as well. And one thing that we looked at was whether or not we could use a G9A inhibitor, and would that have any effect on adiponectin. I don't know what you guys know about adiponectin. Excuse me. But it's an adipose tissue-derived protein, and it mediates insulin action and resistance. You can treat cells with rosiglitazone, and you can increase the expression of adiponectin. Rosiglitazone is an FDA-approved drug. However, it's a very nasty drug, and there are many people that want to remove its FDA approval status. It turns out we can treat cells with one of our compounds, and again, we can show increased expression of adiponectin, and so there's some excitement that perhaps we have a path that we can develop a type 2 diabetes drug for. The other program that we're working on, again, is this DNMT1 inhibitor, and the goal is to develop therapies for acute myeloid leukemia in the elderly. Just a few things about AML. AML is in very serious need for new therapies. There are approximately about 10,000 new cases in the US each year. This incidence increases with age. I don't know if any basketball fans are here, but Flip Saunders, who was a coach of the Timberwolves, he died from AML at a very young age. And not to change the subject, but he did call me to play for his first college that he ever coached at. So. Obviously decades ago. Typically treatments consist of chemotherapy. There are remission rates with this chemotherapy. About 70% of patients do go into remission. However, most of these elderly patients can't withstand chemo, and relapse options are really very limited. Median survival rates typically is less than a year after relapse, and as little as 5 months for older people. There have been some therapies that have been in clinical trials. Decitabine in 2012 was not approved in the US. 2014, cyclacel had a dramatic failure. And so there really is nothing out there on the horizon, and there again is huge high medical demand to develop therapies for AML. One of the things that has been realized by us and others is that when a gene called nucleolin is expressed, this gene, nucleolin, is elevated in all leukemia patients, and nucleolin deregulation contributes to leukemia cell expansion and the progression of the cancer. There are patients that have low nucleolin levels, and those with low nucleolin levels have better survival rates. And it turns out nucleolin overexpression correlates positively with DNA methyltransferase 1 messenger RNA. So the idea that we're thinking is that if we can develop DNMT1 inhibitors and use those inhibitors with patients that have high nucleolin levels, that this will be an appropriate therapy for patients with AML. So again, we've been developing these DNMT1 inhibitors. This is just a short list of some of them. When we look at the activity of DNMT1, when treated with these compounds and compare them to RG108, RG108 inhibits DNMT1 activity by about 15%. Right now, all of our compounds are inhibiting in the roughly 80 to 97% rate. So these are very exciting to us, and we're using these compounds to move forward and hopefully get 1 or 2 of these in clinical trials. One of the things that's really interesting is that our inhibitors are very distinct from 5-ASA. One thing we can demonstrate that when we treat our cells with some of our inhibitors, we can actually upregulate certain tumor suppressor genes. So in this case, lane 3, we're seeing upregulation of CEACAM6 compared to untreated controls. 5-AZA also does this as well, but as we get upregulation of CEACAM6. Similarly, CCND2, both are upregulated with treatment of 5-AZA and with our compound compared to controls that are untreated. But what's interesting, after about a week of treatment with 5-AZA, all these cells die. In contrast, when we treat them with our compounds, the cells aren't dying, but the tumor suppressor genes expression levels are being maintained. And we think that that's hugely important and gives us an opportunity to treat cancers in a manner that would be consistent with a reprogramming event. So just some of the advantages of our inhibitors. It's a completely different mode of action from what's out there right now. Again, our inhibitors are SAM analogs that compete directly for the SAM binding site. They're non-nucleoside inhibitors, that is, they don't lock the enzyme in place and potentially make them much less toxic than things like 5-AZA and 5-AZA derivatives. And we think that ours are going to be much more potent. We're currently getting them into the very low nanomolar range right now, and that's progress that we're proud of. Again, this is our timeline to get our compounds into clinical trials. Again, we think we can get an IND submitted late 2019. Our estimated costs are around $3.5 million. And as I mentioned, we've secured $7 million to drive this in that direction. And so that's kind of it. I hope we got something out of that. I know Thanksgiving's coming up, so I thought I'd just show everybody happy Thanksgiving. And I'm more than happy to answer any questions. Yeah.
What kind of animal model do you use for a drug like this?
So that's an interesting question. So there are a series of models. Okay, typically we'll start off in mouse models, and we actually don't do that here. We'll use a contract research organization that has the models and is used to doing those, and they'll generate the data. Eventually they have to go into a dog or something like that. So typically we need two models before we can justify an IND and go into the clinical trials. And again, we don't do any of that here, or wouldn't do any of that here.
What type of compounds are formed that makes the drug so toxic? And is it how it's metabolized, or is it reacting with something?
So what happens with DNMT1? So DNMT1 goes and interacts with the DNMT1 enzyme. And basically what it does is lock that enzyme in place on DNA so that you can't get any further methylation, for example, when the cell's going through cell division. And the problem is it kills cancer cells, but because it gets into all other cells too. The same thing happens in other, when cells divide quickly, intestinal cells, all that kind of stuff. And so it just becomes really, really harsh for the patients. Does that make sense? Mm-hmm. So the compounds that we use don't allow for that kind of locking in place of the enzyme.
It's fine, a different kind of question.
Yeah.
What would you say, or what can you advise for a new investigator who's interested in doing similar kind of work to what you do, or how you started it, that has a typical faculty position in a university?
So say that again. I want to make sure I understand your question.
Right. I'm asking about a new faculty member who's interested in doing work typical to yours, where you're looking at I guess in a tenure track position.
So, I mean, it's the same, it's a nice, very real professional plan for graduate students to postdocs to junior faculty, and obviously the mentorship is the most important part about that, right? So, if there's a certain path you want to take, obviously your postdoc mentor probably is the most important person. Mm-hmm. And one of the disadvantages I have here is that I don't do nutritional work, and so in terms of the colleagues that I have here, there's no one really to work with in terms of what I do. And so when you're looking for your faculty position, and you have something in mind that you want to do, you also have the people around you that can benefit you as you go through that traditional NIH line. It's a standing line process, obviously.
So in the process, you're making discoveries that you publish so they become public?
Absolutely, yeah. Publishing is important no matter what you're doing. So,
okay.
I also consider patents an important. It's equivalent to a published paper. Maybe harder because it takes a lot longer to get it approved. But never, if you really want to see what's going on out there, go to the patent office and do searches in your field. Because a lot of that stuff will be there, but it won't be in published documents, because, for a lot of reasons. But
Okay, yeah.
I don't know if I answered your question.
Yeah, no, we have.
Yeah.
At least, you know, we have young professionals here and
Yeah.
just,
I would say, though, that if anybody thinks that you're going to be completely funded for the next 50 years of your life, you may want to think about that a little bit, because funding goes up, down, and you can lose funding. I think it should be creative in terms of sort of the resources that you may go after, because there are other resources out there that just standard NIH and SF grants. So.
and it kind of happens no matter what you're working in.
Right,
It's
right.
I mean, something's hot now and,
Right.
you know, it's,
Hi.
What type of a specific cells they use when they clone Dolby, I think it was the name?
They use a mammary. They use breast tissue.
Because I hear that if the animal gets sick after, when it gets sick really fast.
Yeah, so the thinking with that, so there's no such thing as a perfect clone. And when you reprogram a nucleus, you don't erase the entire epigenetic memory away. There's always some of it that persists. So there's kind of a tug of war between sort of maintaining certain functions of the somatic donor cell and then being reprogrammed to, say, a pluripotent state. And because of that, cloned animals typically are associated with a lot of diseases. And that's when it became really realized that the epigenome contributes to disease, okay? Because this epigenome wasn't being completely erased and there's some persistent memory that always hung around. With Dolly, the problem with Dolly turns out to be that Dolly became a pet and she was overfed, she was overloved, overpetted, and Dolly died early. The thinking really is Dolly died early because she was treated more like royalty than a sheep, okay? But probably if Dolly lived a full life, you would have seen other things manifest that you just necessarily wouldn't predict. So like virtually every cattle that we had cloned eventually had arthritis. And we were producing therapeutic proteins in their milk, but their arthritis was so bad that we actually had to have things to help hold them up, okay? And that was totally unpredicted. And there are other events as well that were very common. So the notion that you erase the epigenome and you start all over is not really accurate. There's a persistence.
I have another question. So what is the difference when you said that you develop a disease and it's genetic disease or is because of epigenetic factors?
So I was trying to give you a sense. So there are a lot of diseases that you can give a genetic reason for, some sort of mutation, okay? In cancers, there certainly are genetic aberrations that contribute to cancer. However, if you look at sort of the number of aberrations that are genetic, and then compare them to the potential number of aberrations that could be epigenetic, the epigenetic aberrations really outnumber the genetic ones at a ratio of around 10 to 1. So there's just a lot of the take-home message, there's a lot of opportunity for things to go wrong at the level of an epigenome. And that's why people are interested in targeting it for diseases. Because there may be more areas that you can work with a little bit more efficiently, potentially.
What do you know then about in vitro fertilization?
Say that again?
What about in vitro fertilization? Are there things that can go wrong there?
Yeah, so there's actually been colleagues of mine at Penn actually looked at in vitro fertilization and culturing embryos in vitro. And it's very clear that that can affect epigenetic programming. I mean, one area that they looked at were imprinted genes, for example. And they could show that embryos that were cultured in vitro had different methylation patterns on imprinted genes. But the long-term consequences of that don't really know. Obviously, in terms of humans and IVF, there's a huge experiment going on right now. A lot of in vitro fertilized babies, but you know, a lot of these babies seem to be doing just fine too as they grow up. I think the first in vitro produced person was in the 80s, and so they'd be.
And so you don't know what's going to happen when they're 50, 60, 70 years old.
But truthfully, you don't know that anyway,
Oh.
in general.
That's true.
I'm 58, so my wife and I were talking about, oh, we've grown our kids, we have these grandkids, life's good, but now we're entering the phase where, man, things can really go wrong, okay? And so we try not to think about that.
You know too much.
Yeah, so, yeah.
Have you looked at the compound that you're looking at? What's the LD50 for just home or something?
So we do LD50s, we do total inhibition, and I don't have those numbers in front of me, but we do generate those numbers. I'm in L2020, if you ever want to stop by, I can dig that stuff out for you. But you know, it can vary, compound to compound, cell type to cell type, that sort of thing.
How much better is it for the, comparing to the drug that's being used right now? That you said was-
Oh, our LD50s are better, so that much I can tell you, yeah. Any questions?
I guess what I wanted to ask is in relationship to, I guess, epigenetic modifications, is it better to, I guess, kind of treat the problem like a drug or maybe just modifying the environment? And would that be, I guess, like more efficacious?
Well, for cancer, I mean,
Just
you have
treating the drug?
typically for cancer, you have an endpoint. I mean,
Right.
you're trying to avoid that endpoint.
Mhm.
So things like diet and lifestyle changes, those are long-term events. So you're not going to be able to treat cancer just by diet alone, stop smoking, for example, at that stage. But there's some interesting work looking at methionine restricted diets in combination with chemo that seem to have an effect. I think in terms of, you know, lifestyle, that's a generational issue, and it begins even before you're born. And I think that that's the knowledge that people have to understand, that it's important to take care of yourself, it's important to take care of yourself before you get pregnant, and it's important to get your kids on the right lifestyle path too. Problem is that, you know, we're all teenagers at one time, we do dumb things. But, did I answer that?
Yeah, I guess I'm just kind of, I know like you can't go back and, you know, change things that have happened, so I'm guessing the thing now is to like treat these things with drugs, but I guess is there a way that we can kind of start where we are and try to work on like changing things in our environment, or is it just at the point where we need to use drugs to treat?
Well, I think, I think
diseases?
I have an answer for that. So one reason I went into cancer, because when I came here, I did make a sincere effort to try and sort of look more at nutritional things with regards to the epigenome. And I found that really impossible to do. I mean, it's hard to do. It's hard to design those studies, even with mice that turn over rapidly. As hard as cancer is, it just seemed like, you know, you can make an impact in my lifetime and in a generation. You know, how do you preach to the world about preventative medicine and living healthy lifestyles? I mean, we have been preaching that since before you were born, for example, and it hasn't necessarily manifested well. But at the end of the day, healthy lifestyles are the best thing, period. I mean, I prefer not to have a drug. I hope my mom didn't smoke when she was carrying me and drink, you know, those types of things. So, I don't know if I'm answering your question, but it's a good question. I mean, it's a really important question.
I also have another question. As far as like the different epigenetic modifications that are a result of like drug treatment, have there ever been like any tests on like the long-term effects of those over generations?
You mean in terms of drug treatments? No, because there haven't been that many, there aren't really many epigenetic drugs out there right now. But the question you're asking is that if you treat a person with an epigenetic drug, can you induce other pathologies? And the likelihood is very much so, yes, because unless you can target it specifically to what you want that drug to hit. And a couple of us are actually working on that, so there are potentially ways to target a drug to, say, a specific gene in the liver, for example. Say you have GF21. Obviously you have to deliver that drug specifically to those liver cells and not allow it to get into the other cells. So I can't really answer your question, but in terms of the possibility that you introduce other pathologies, that is a real danger in terms of epi drugs. Now, if you're a cancer patient, especially if you're in stage 4, you really don't care. You know, if you just want to survive and have a high quality of life, right? So, but yeah. So one thing that I was criticized on, and I don't know how, I was given a talk somehow the notion of giving an epi-drug to a pregnant mother came up. But you would never want to do that, right? Because the child's pregnant, and that's probably a very vulnerable time in terms of how DNA is getting programmed, especially very early on. So, but it's a good important question. So thanks.
Other questions?
So just to let you know, our compounds are a very low nanomolar range, okay? And so 5-AZA actually works at micromolar range. And so one reason that our LD50s and these other types of parameters are better, we think it's because of that tight binding that we have.
That's all, it's really interesting.
Well, good, I hope you got something out of it, so.
Yeah, thank you very much.
Thank you.
So I was at the University of Connecticut, and Jerry Yang.
Oh, yeah.
The cloning happened about the same time
Right,
as the cow,
right.
right? And
Yeah.
He didn't live that long after that,
No.
unfortunately, but
No.
he was right next door to me.
Yeah, no, he's a good guy.
You knew
He's done
Jerry.
very academic work, okay?
Yeah,
And,
yeah.
I think he even claimed that he did the first.
He did,
Yeah.
he did claim that, and
Right.
and that's what I
He had the
remember, because
first-
I was right
He had the
there.
first published one. He's one
Okay.
of, we couldn't
Is
publish
that right?
ours at that time because
Okay.
we were getting involved in a huge patent lawsuit.
Okay.
So we were being, it was a three-way thing. Geron, ACT, and Infigen
Okay,
were all in this big, huge fight.
Yeah.
Everybody lost, okay? So we couldn't publish our stuff, but it
okay.
was,
But you
yeah.
did the work.
Oh, yeah, absolutely,
Yeah.
yeah. And I was very sorry when he died so young, he was way too
Yeah,
early.
yeah,
Well, not so young, but he was way
was
too
young.
early.
No, he was young, he
Yeah.
was young. His son was in my youngest son's class in high school, junior high at the time. But he took, I don't know, it was sort of Jerry's junket, he took a bunch of us over, the dean supported it.